Hey guys, happy Wednesday. Welcome back to Fiveable's AP Bio Streams. My name is Jessica Nadzam. I am your host. In today's stream, we're going to be talking about enzyme catalytics. So super glad you're here. I've been teaching AP Bio for about 5 years now, so I like to think I know what I'm doing. And I'm also an AP reader, which means they take me to a city, they lock me in a convention center, and they make me sit in a chair for 8 hours and grade your tests. But I learned a lot about, how to grade AP exams, which makes me good at teaching them. So it's kind of helpful. It's a win-win. We have Twitter, Instagram, YouTube. We have like a lot of stuff on the internet. And we're millennials, so we like it when people follow us and when they like our content. So if you could do us a favor and just like hop on your phone later when you have a minute and like that stuff, that would make us really happy. So yeah, that's the main thing. Please go like all our stuff. They may even like you back. If you post anything about Fiveable, we will definitely give you some brownie points in some way. I don't know what way, because I don't run the social media accounts, but I have heard rumors of great things. I will leave it vaguely there. Just please follow us. You know what I mean. Okay. In this stream, what are we going to do today? Here's our little outline. We're already 5 minutes deep, and I haven't actually given you any content. That's probably what you want, huh? So talking about enzyme catalysis today, but before we can do that, we need to understand the things that will help you understand enzymes. Think of it this way. It's your very first day of math class, and the first thing your teacher asks you to do is long division math, but you don't know how to add, you don't know how to subtract. You're in kindergarten, you know numbers 1 through 10. I would not ask you to solve a problem if I didn't know that you had the prerequisite knowledge behind it. Now, I'm not saying you guys like, oh, you don't know this. What I'm saying is a lot of you probably learned this at the beginning of the year. That was 9 months ago. And our brains have gone to sleep a couple times since then. And sometimes it takes a minute to refresh that information. So my goal today is to refresh that information from you. I like to do lots of Q&A, so make sure you've got that chat box pulled up so that when I ask questions, you are ready to go and answer and be the first one, because I know that's very, very important to all of you. But to understand enzyme catalysis, we need to understand biomolecule structure, specifically proteins. We need to understand enzyme structure, and then we're going to get into the FRQ section. So someone asked, do you have any study tips? Honestly, the thing that got me through high school and college, especially in sciences, was I would take my notes, and if they were handwritten, I would type them up, and then I would write them out by hand again. And if they were typed, I would write them by hand, and then I would type them again. That's literally all I did. Occasionally, I used Quizlet and things like that for content-based things. But in order for me to truly grasp information and know how to apply it, which is what AP is, I had to understand things backwards and forwards. So I would use flashcards in the beginning, but then really just rewriting it was what got the stuff in my head so that if someone asked me a question, I could think it so quickly that I had enough time to do the application part. So first steps first, memorize it, but then you got to go past the memorization. Someone says it's the pollen. Yeah, Sierra, it is. My car is yellow. It's not supposed to be yellow. Can you send us the slides? I will release them at the end. Is there FRQ practice at the end? Yes, I have a practice FRQ that we're going to analyze, and it is based off of the AP format. So, woohoo! Oh, there's more people now. We have like 16 people. It's a party. 16 people feels like a lot for me now because, the only person I see is my husband and my 2 dogs. I like my husband and my 2 dogs, so I guess that's a good thing. Okay, let's go ahead and do some content because that's what you came here for. First things first, let's talk about biological molecule structure. This says biological structure because, whoopsie, when I was typing this, I was in a hurry because, we're all in a hurry, aren't we, to do something. So biological molecule structure review. Also sounds really wordy now, but whatever. So there are 4 biological molecules. They're also called biomolecules. They're also called macromolecules. We have proteins, carbohydrates, lipids, nucleic acids. Those 4 are going to be the building blocks of life, okay? Each of them has a very, very specific job. For example, what is the job of carbohydrates? Energy. Very good. Is it like quick energy or like long-term energy? Short-term energy. Very good. Okay. So if carbohydrates are like your quick short-term energy, like propane or something, what would be your biomolecule that's like diesel? It takes a long, long time to break that stuff down. Lipids, excellent. Lipids are going to be a lot harder to break down, but they are a source of long-term energy. Very good. Where do we store our genetic information? In the nucleus, but in what nucleic acids? Very good, Ida. Yes. So we store them in the nucleus, we store them in DNA, but in the nucleus, we have nucleic acids, which make up DNA. So very good. Okay. And then finally, what does all the other stuff in the cell? What biomolecule does all the other stuff in the cell? Proteins, yay! Okay, so we're going to focus mostly on proteins here, just so you know, for the rest of this conversation. You need to know what the other stuff is, obviously. So I wanted to do a quick review of it. But the main thing we're going to talk about here is proteins. So what are proteins even made of? Amino acids, very good. So if I go in here, I wonder if it'll be nice today. It's probably not going to be well. So we've got our amino acids. You've got this H2N. That's an amine group, like amine, like ammonia. Ammonia is something that smells not that great, but it's used in a lot of cleaning products. It also comes from a lot of animal waste. Amine groups, this NH2 or ammonia, amine, ammonia, that's how I remember it. That's a critical part of each amino acid. Okay. Then you've got this OHO with double sticks, or as the scientific people tell me I'm supposed to call it, a carboxyl group. You have to have both of these. And then you have this R group. What is the significance of that R group? I know, so mean. Variant. Okay, tell me, what do we mean by variant? Like what does that mean? The R group decides the type of amino acid. It's different for every amino acid. They can change the polarity and determine the properties. Perfect. Oh, I'm so smart. Yes. So the way that I explain it to kids who haven't had this before, do you remember that day in like 5th grade when they put letters in math? When they would be like, find X, and you'd be like, there's not supposed to be an alphabet in math class. Basically, if in math you're always trying to find X, R is like the science version of X. That's the thing that can be interchanged with anything else. In math, X can be whatever. You have to go find it because that's math. They say, go find the X. And you're like, it's not my job. But then the teacher's like, yes, it is. In science, you don't necessarily have to find the R group. You just have to know that there are like 20-something different possibilities of R. Complicated, but the reality is you don't have to memorize those, so that's not really your problem. Like apparently back in medieval times, aka the 90s, they would like make people memorize all of the amino acids, which I'll be honest, I don't think I would have made it through that degree. I can't rewrite all of those enough to remember them. So instead, we don't have to do that. We have charts and stuff for that. Should we know the groups? Like, yes, I would know the structure of the amino acid. So like the amine group and the carboxyl group, but those will never change. Those will always be the same thing. The R group, it just changes for every different amino acid. Do we need to know all the functional groups? So here's what I tell people, because I teach my students functional groups, but I don't like test them over them. It's just kind of like we do it as an activity, like at the beginning of a unit, just as kind of an icebreaker. But the reason I tell my students about functional groups, I consider functional groups to be like special ingredients. So here's how I explain it. If you've got, hang on, I need to post it for this very official demonstration. So let's say, also I'm an artiste, so don't laugh at this. So let's say you've got a cupcake, a plain cupcake, no flavor. It's just like white cake. It's like whatever. Okay, that's fine. So think of a molecule like a plain cupcake. It's boring. It's just a bunch of carbon and hydrogen. There's not much on it. But on a cupcake, sometimes I can add cool things like chocolate chips. And that gives it a really sweet, special taste. Well, what if I told you you could add an OH group to that boring molecule? OH, that's a hydroxyl group. That's a functional group. And that would make a molecule basic, as in like soapy. Okay? What if I told you on this cupcake I put, what's another topping? Sprinkles. Okay, sprinkles don't really have any taste, but we like them for some reason. Texture, I guess. So we add sprinkles to our cupcake. So we have sprinkles. Okay, sprinkles are a different, totally different thing you can add. But it'd be kind of like if I added a totally different thing to my molecule. Like, let's see, what's another one that I could add? Like an amine group. An amine group, which would make it ammonia, kind of acidic, things like that. Okay. I teach my kids functional groups because eventually they get familiar with the functional groups and know that, oh, an amine group means it's probably involved in this. A carboxyl group means it's probably involved in this. What I will tell you is you don't have to memorize those to do well on the AP test. But if you're someone who already has everything memorized, it wouldn't hurt to familiarize yourself with them just because it'll help you go through the stuff faster. But there is nowhere in the AP Bio curriculum that says you have to know it. I just teach it to my kids because I'm extra, as they tell me, like every day. Okay, very good question though. So we've got our protein, it's got amino acids, it can be all these different combinations. Quick summary here. I love tables. It just helps me like organize my thoughts because if you haven't heard from like my rambling in the past 15 minutes, my thoughts are not always the most organized. That's why I need super detailed, GIF-filled PowerPoints in order to like keep my focus. Tables make it very, very easy. Whenever you can draw a table in your notes, that is a great way to organize your thoughts because we know that all of these 4 molecules are going to have a certain structure, a certain function. And if you can organize that in a table and you draw that table, like say 3 times over, it sticks in your brain pretty well. It's a sort of, I wasn't a psych major, but I took several psych classes and it was some sort of like spatial organizing, which is supposed to be good for making connections in your brain, which means you remember it. Just draw a bunch of tables, y'all. It'll help you remember stuff. So carbohydrates, they're made of monosaccharides, polysaccharides, glycerol and fatty acids, nucleotide, Again, I'm not so worried about the other 3 for this particular stream. I'm more worried about proteins. So we know that proteins are made of amino acids. You string them together, you get a polypeptide. Their function is work. They can do all sorts of stuff. They're literally anything. They can be for structure, like building the walls of a cell. They can be for reactions, like we're going to see in a moment with catalysis. They can be for signaling. I mean, protein does all the cool stuff, basically. So, woohoo, yay. Does anyone have any questions about biological molecules so far? I really need to sneeze, so if I sneeze in the middle, I'm sorry. Any questions? The 4 stages of protein. Oh, you mean the 4 levels of structure? We're going to get to that in just a moment, but that's a very good question, Talia. Which ones are polar and which ones are non-polar? You won't need to know that for the AP exam because carbohydrates can be polar or non-polar. Lipids can be polar or non-polar. They obviously have areas where a basic carbohydrate is one, a basic lipid is the other. But sometimes you get things like, say, glycolipids, which are a carbohydrate and a lipid put together. So I wouldn't worry about memorizing that for the purpose of this exam. You won't need that information. If you do, it will be given to you on the test. Does that help, Juliana? Now, someday, if they ask you, what is the polarity of a lipid, you have full permission to leave me a negative 5-star review on Fiveable, because that would be my bad. But I really don't think that will happen. Now I'm going to have bad dreams about negative 5 stars. Okay, now we're going to talk about protein structure. So like I was saying a minute ago, proteins do all sorts of stuff. So they're involved in contraction, that is with your muscles. They're involved in hormones. They're involved in your immune system, transport, enzymes. They store stuff. They are involved in structure, and they also have receptors. Again, they're based on this model with, what is this thing that I am circling? The purple thing. What is that? What's the purple thing? Tell me, tell me, tell me. The amine group. Very good. What's the one on the right side of the molecule? What is it? What is it? What is it? The carboxyl. Very good. Okay. And then we've got this central carbon, and then we've got our R group, and that can be anything. And depending on what it is, we'll give it certain characteristics, certain properties that it'll let it do a certain task. So going forward, keep that structure in mind, because then we're going to get to Talia's question about protein structure. My kids hated this at first. They were like, why do I need to know the tertiary level of a protein? And I was like, you will see. And you guys will see in just a minute too. You also learned it in gene expression, which I gave a lesson on like a month ago. Go look it up. Basically, proteins have multiple levels of structure, kind of like humans have multiple levels of structure. Any living thing has multiple levels of structure. So I'm going to give you a little example, or a couple little examples. First one's a plant. Ignore the fact that the plant looks not good. It was recently transplanted because I had a big plant, but it didn't fit in the pot. So I pulled it apart into some little plants, and the big plant is doing fine, but the little plant, I don't know if he's going to make it. But the point is I got a plant. I also have, pretend this is a real otter and not a tape dispenser. These are the things you find in a biology teacher's house, plants and fake animals. So any living thing, whether it's a plant, a real otter, not a tape dispenser, or me, we're made of cells. That's our basic structural unit. What do all those cells come together to make? Tissues. What do all the tissues come together to make? Organs, and all the organs come together to make organ systems, and then the organ systems come together to make an organism. Yay. Okay. Proteins are the same way. Proteins start out as an amino acid. And all of the amino acids come together to form the primary structure of a protein, which is called a, what is it? What is it? What is this? What do we call that thing? A polypeptide. Yay, Juliana, good job. Okay. So the first thing is a polypeptide chain. So all those amino acids come together to form a polypeptide. But then, because all of those R groups are different, why is my face still so big? I'm sorry. You don't want to look at that. You want to look at the slides. So, but if you remember, each amino acid is different. Okay? So, depending on the different amino acids and the different charges of the R groups, the polypeptide is going to fold and bend and shape and sliver. And it can either become a beta pleated sheet or an alpha helix. So it's either going to... So this is the tape measure I always use to explain this problem because it looks like a polypeptide. It just works. I'm going to make my face big again. Sorry, guys. That way you can see this. So it can either be a beta pleated sheet, which is kind of like... Let's see if I can do this facing you guys. So like if I was to pleat this kind of like an accordion or whatever, and that would be because there are certain bonds in the R groups that are making it bend a certain way. Now, on the other hand, there can be different bonds that make it, instead of pleating, it may coil. Woo! And so it's nice and round. Whoops. Woo! Okay. So it just depends on the charges in the R groups in terms of what structure it's going to be. But then we have the tertiary level of structure. And that tertiary level is when it starts to bind and bond even more. And as a result, you get this clump, this little like coiled up ball. If you've ever seen like a 3D image of a protein, they never look like Pac-Man. Like I always see them in lectures as like Pac-Man with like, oh, pizza cut out of his mouth or something. And I'm like, that's not actually what happens. It actually looks like this, like this coiled little ball thingy. That is what a protein looks like. Just a bunch of strings all coiled up around each other. So you get it all coiled up. That's tertiary structure. But sometimes, do I have another tape measure? I don't think I do. Sorry, I've only got one tape measure. But sometimes that protein will bind to another protein, and that's when you get quaternary structure, when several of them come together. This is very, very important because think about it. If you change a single R group, will that amino acid have the same charge? If you change a single R group, will that amino acid have the same charge? No. Very good, Soleil. Very good, Japesh. Okay. So as a result, is the protein going to fold the same way? If it has a different charge, is it going to fold the same way? No. Very good. And that will make a different protein. And if a protein is in a different shape, can it do the same job? Nope. So it's very important that this structure is correct. That's the point I'm trying to make in this. So hopefully I've stressed that enough to you, and we can move on to enzymes. So like I said earlier, proteins are not like happy little Pac-Man like I'm really sorry. They don't look like this, where you see it looks kind of like a Pac-Man. It's just got a little piece sticking out. That cartoon representation is fine for biology as long as you understand it doesn't actually look like that. It looks like this, and there's something that fits in everywhere. Just remember, change in structure equals change in function. Very good, get beat. Okay, so you've got this super specific, wiggly, writhing protein structure. It's got all these bonds that are constantly going, okay? An enzyme is a special form of protein. It is a protein that is tasked with making reactions occur faster. Many of you may be familiar with the American Treasure film franchise called Fast and Furious. If you are a driver in Fast and Furious, what do you put in your car to make your car go zoom, zoom very fast? Gas, fuel. Nah, in Fast and Furious, they put something special in it. It makes like flames come out. Very good, Juliana. Someone has been paying attention in movie class. Very good. NO2, they call it NOS. They put nitrous oxide in their cars, which pretty sure is bad for the environment or it's illegal somehow, but it's a Vin Diesel movie. So like, what do you expect? So they put this NOS in their cars and it makes them go super, super fast. Enzymes are the same way. They're kind of like NOS. They're what makes the cars go zoom, zoom, zoom super fast. It makes your cells go super fast. So what happens is, you can see down here, okay, you've got your enzyme. That's this gray Pac-Man thing that is not anatomically correct, but you get the point. That enzyme binds to a substrate. It has to be the perfect shape. So remember what we just talked about. If the wrong R group is used, is the protein going to have the same shape? No. So is the protein still going to be able to react with the substrate? No, it's not going to work anymore. It's like a lock and key that won't fit anymore. And that's not good because then you can't do the reaction. You can't go faster. Okay. Normally, the enzyme and the substrate go together and the reaction can happen very, very quickly, which is good. A lot of our reactions must happen quickly. Think of it this way. You're probably sitting in your bedroom or your kitchen or somewhere on planet Earth. And while you're sitting there, what if you looked up and the door swung open and a bear walked into your room or house or whatever? What would you do if a bear walked into your room? Throw a chair, attack it. I feel like the bear question is like always a personality question, finding out people who would fight or flight. So, okay, would you wait like a couple minutes to do that? Like kind of scratch your head and go, it's a bear. I wonder what I should do. Or do you make that decision instantly? You do it instantly. Very good. Okay. That's because you have enzymes and substrates in your body that instantly can snap and react, and you can go and either fight or flight. Whatever you're going to do with that bear, that's your choice. National Park Service recommends making a lot of noise and playing dead, but I'm not like a park ranger and I'm not like a bear master, so I don't know what I would do either. Point is, those enzymes and substrates have such specific shapes that they can react so quickly. And that's what keeps you alive in stressful situations, whether it's your body reacting to a negative substance, or it sees a bear, or even just your blood sugar, your temperature, and your blood pressure having to react to all the stresses that we put our bodies through. I'm sure some of you do sports. What happens when you start running around in circles on a court or a field after a while? Lactic acid, what else? Cramps, sweat. What about in your blood and your heart? You lose energy. Tell me about your blood and your heart. Your heart rate goes up, okay? Your heart rate can go up, but that is a stressor on your body that is out of homeostasis. So you have to have an enzyme that once you stop running around, you can chill. And it can tell your body, okay, we all good now. You have to be able to do that quickly or you cannot survive. Since you're all still sitting in front of me, I think you're good though. So enzymes are very, very specific in what they do and how they do it. What they do is they lower activation energy. So for example, we've got two lines, a red line and a blue line. We've got the amount of energy it takes to do a reaction. And then on the bottom, that thing will go away, we've got the reaction. So if I don't have an enzyme, let's say, for example, I just ate some ice cream and I need to break down the lactose in that ice cream, okay? Now, if I don't have an enzyme for that, say lactase, it takes much more energy to break down that lactose. That's a lot of energy wasted, which if you remember in biology, humans or any organism are only fit when they survive and reproduce. And to do that, they need to conserve as much energy as possible. We're kind of like energy hoggers or energy hoarders. Every time we expend unnecessary energy is technically a problem. Like, we're not supposed to do that. We're supposed to conserve it all for surviving and reproducing. So if you're eating food that takes more energy to digest, that's not considered like evolutionarily fit. But if you're eating a food and you have an enzyme, that's this blue line, that makes you digest it without using as much energy, that's considered fit. I want you to think of it this way. Let's say you're in New York City and you can pick between two cars. You can pick between a Hummer and a Prius. Which one will more efficiently get to Los Angeles, California? Prius. Why? Prius because it's fast, it's lighter. Nah, keep trying. Better gas mileage, very good. So like a Prius can get like 5 zillion, I don't know how much, like 40, 50 miles to the gallon. I don't know what the latest one is. It gets places very efficiently, very quickly, and it doesn't need as much gas, okay? However, a Hummer takes much more gas and it doesn't go nearly as far. So that would kind of be like this without enzyme. You'd be spending all this extra energy to get to the same place. Why would you spend all the extra time and money when you could get to the same place for a lower cost, energy-wise? That's what enzyme catalysis is all about. Does anyone have any questions so far? How does it do what? Lower the activation energy, you mean? So enzymes, the reason they lower activation energy is because they break down these things, substrates, into smaller and smaller pieces. Whenever you break things down into smaller and smaller pieces, it's easier and more efficient to basically finish doing what you need with them. Think of like whenever you have a bunch of homework. If you try to do all of that homework at once, your brain might explode. But if you break it down into like 20-minute sections, you can do it a lot more easily and a lot more efficiently. Same thing goes here. We're breaking them down into smaller, more manageable pieces. That's the metaphor I'm going to use for this. Whoop. Any other questions? So this is kind of repetitive. So again, going back to my ice cream example, and this may give you a little bit more context, Sebastian, on my last answer. So lactose uses lactase to break down, and that lactose gets turned into a glucose and a galactose. That means you can now digest it because it's in smaller pieces. So it takes less energy as opposed to if you tried to digest this whole thing all at once. That's why lactose intolerant people get sick. They can't digest the whole thing all at once. So to do this, you've got to have an enzyme and a substrate. They come together and they break the substrate apart. That's all that's happening there. So that was kind of repetitive, but at least there's a baby getting excited about ice cream. So that's cool. Ooh, we're already at the FRQ. That was so fast. So if you were with me last week, I tried to do it one way where I had everything in like PDFs, and that was fine, but it like stressed me out because I couldn't just show y'all everything all at once, and I didn't like that. I want to show y'all everything. So this time I tried to put the question on every single slide and just put the different parts of the questions like A, B, C, D. So bear with me. We're trying a new way. Hopefully this works. But let's talk about the question. So this is our enzyme question that we're going to discuss. An experiment was conducted to measure the reaction rate of the human salivary enzyme alpha amylase. So if you know, in your mouth, you spit. If you didn't know that, I want to get that checked out. But your spit is more than just a liquid. It has enzymes that breaks down your food, okay? So, and one of those is alpha amylase. So 10 mil of a concentrated starch solution and 1 mil of alpha amylase were placed in a test tube. The test tube was inverted several times to mix the solution and then incubated at 25 degrees Celsius. For context, anytime you see 25 degrees Celsius is like, I know human body temperature in Celsius is like 36, 38. So I would imagine that's near-ish room temperature, but it's certainly not frozen. So the amount of product, maltose, present was measured every 10 minutes for an hour. The results are given in the table below. So we got a lot going on here. We've got starch, and we're mixing with amylase, which we know is an enzyme that digests things. In this case, apparently carbohydrates. Remember, we talked about those. So that amylase breaks down the starch, and as it breaks down the starch, what does it turn the starch into? Maltose. Very good. Okay. Just a heads up, in case y'all hadn't already heard this, anytime something ends in this ose, that means it's a carbohydrate. If it rhymes with gross, it's a carbohydrate or a sugar. Anytime it ends in this ace, that means it's an enzyme. So at 0 minutes, we have no maltose. At 10 minutes, we have 5.1 micromoles of maltose. You don't need to know what this unit is. You just need to know, is this growing or shrinking? Growing. Very good. Don't worry about units. Whenever they put like weird symbols in there, do not worry about that. All you need to be able to tell is it getting bigger? Is it getting smaller? That's all you need to worry about. So don't let weird symbols freak you out. Okay. So the first part of this question, identify 2 environmental factors that can change the rate of an enzyme-mediated reaction. Discuss how each of these factors would affect the reaction rate of an enzyme. Okay. If you don't already have it with you, get like a sticky note or your phone, open a note, whatever you need to do. Let's identify 2 factors that can change the rate of an enzyme-mediated reaction. Yes, you will be able to use the formulas, Ida, but you have to bring your own equation sheet. They won't give you one on the test. So make sure you bring your own. Okay. So temperature and pH. I see temperature and pH quite a bit. Okay. How do those factors affect the reaction rate of an enzyme? Denaturation. What does denaturation mean? Change its shape how? Changes in the structure? I need some complete sentences, y'all. There's a lot of like 3-word answers, and I don't know what relates to what. Pretend this is the Relay P test. Discuss how those 2 factors could affect the reaction rate. Change the shape of the activation site. What changes the shape? Complete sentences. Capital letter and a period. Temperature. With a higher temperature, they are able to break down faster. Change the temperature and pH. Okay. You're all giving me the answer. The enzyme can no longer interact with the substrate. Okay, good. You are all giving me pieces of the answer, but I will warn you, if you don't give all of that information in a complete sentence, they're not going to give you a point for that. So the pH disrupts the bonds in the enzyme, causing the active site. Okay. Change in the tertiary structure. Very good. Okay. Much better. Period T. Yes. Okay. Much better on the tail end there. Good. Identify, you just have to list it. So like 1, 2, that's it. But anytime it says anything like discuss, explain, you need to give like a 3 to 4 sentence response, to be honest, or a 3 to 4 line response. Okay. Otherwise, you're probably not giving enough information to get the points. That's just the rule I've seen so far whenever I grade papers. So let's check the answers. Identify 2 factors. You could put temperature, pH, salinity, inhibitors during mixing, pressure, oxygen, light. Very good. We got that. For temperature, you could have said temperature increases the rate, temperature decreases the rate, high temperature causes denaturation. That would have been good. Some of you got a little more specific with the improper folding, the change in the active site, all those things. Any of those would have been fine. So can you repeat your rule, please? What rule are we speaking of, Talia? I have lots of rules. 3-sentence discussion. Okay. Typically, if they tell you to discuss or explain something or describe, you're going to want to do like a 2 to 3 sentence response, if not a response that is like 3 to 4 lines. If it's just like 1 sentence, it's probably not long enough, because think about it. A discussion, you can get up to 3 points. Usually it's 2 points, but up to 3 points. Think of every sentence as being worth a point. Denaturation does not change the sequence of the protein. Very good, Juliana. So when you denature things, yes, the protein breaks down. It unfolds. So like if it was furled up in this little ball. Denaturation means it loosens, and now it's not the same shape, okay? But the bonds and the amino acids are the same. They're just not squished together. Denaturation just means it all kind of fell apart. Okay. All good so far? Okay, cool. Let's take a look at part B. So now you're going to explain why a change in the reaction rate was observed after 30 minutes. Why does it change after 30 minutes? What do we think? Remember, complete sentences. Explain. So it's probably worth 2 points. Probably going to need 2 sentences. I'm going to drink some Gatorade and let y'all compose your answers. All of the substrate was being acted on by the enzymes. Active sites were all engaged, so the reaction rate leveled off. All the substrate was used by the enzyme. There was no substrate left. As time goes on, less solute will be available to be bound to. Enzyme's active site from the enzyme-substrate complex was starch and catabolized into maltose by lowering the activation rate. Enzyme maximum. Good. Yeah, this looks great, guys. Change in the reaction, catalyzer breakdown, starch. Okay. So Laura, Callie, these are both really good responses. Ida, this is good. I'm going to warn you. Ida, what this one looks like is you're kind of regurgitating what it's doing. You're not saying why it's doing it. So you're telling me how an enzyme works. You're not telling me why the enzyme is leveling off. So you'd probably get partial credit for that, but you need to put the part about as time goes on, there's going to be less solute available because it all gets reacted with. Okay? So these look good. The actual answer key says reaction rate slows or level off. Ooh, this is 2 points, like I said. So you get a point just for saying the reaction rate slows down. And then another point for saying it slows because substrate concentration declines or the substrate is used. Or you can say the enzyme is inactive by about 40 minutes. Ooh, either one of those is good. Soleil, that looks good. It's totally okay, Ida. Like, seriously, we all do it. That's why we're practicing now. So don't think I was calling you out. I just, while you're here, I want to make sure you're getting the best information that you can to improve. So no worries. Okay. Anyone have any questions about part B? Now we're going to draw and label another line on the graph to predict the results if the concentration of alpha amylase was doubled. Explain your predicted results. So one thing I just realized that I forgot to do, I didn't put the actual graph in here, but this is something they may ask you to do. They're not going to ask you to construct a graph, but they may ask you to add something to it or interpret it. So in this graph, what we're going to see is that as time goes on, whoop, that's not what I wanted. As time goes on, more maltose is going to get used up, and then it's going to level off. It's going to plateau. Now, if we add more amylase, what do we think is going to happen to the reaction? What increases? Remember, sentences, capital letter and a period, y'all. Rate of reaction will increase. The reaction rate will increase. Good. Explain your predicted results. Why will the reaction rate increase? Why will the reaction rate increase? Because there's enough starch now for all the enzymes to be at work. Keep in mind, we're adding amylase, not starch. Japesh, are you talking about the second question or this question? The second one. Oh. No, that would be right. That would be accurate. Okay, no worries. So these look good. Y'all are good. Let's check our answer. Okay, so if we drew a line on here, so the original line would be this one. I'm tracing over the middle one. Okay, the new one, you could either do one down below or you could do one up above. It sounds like most of y'all did above because you said increase. So now your explanation, you have to talk about it. So the substrate is consumed more quickly because twice as much enzyme is present. Yep, I see that from a couple of you. Or you could say more product is formed at each time point because twice as much enzyme is present. The product formation levels off as the enzyme loses activity. That would be if you put line 2. That's the other one. So Yeah, good job, y'all. Y'all did good on this question. Are there any websites besides the College Board website that offer practice FRQs? I mean, I know they go over some on other websites and things. Obviously, I'm not an expert. Most of my work is with Fiveable, but I would imagine that if you Googled AP Biology practice FRQs, you could find some stuff. Non-competitive inhibitors. Okay, so there's competitive inhibitors, which will block the active site. And then there are non-competitive inhibitors that don't block the active site of an enzyme, but they will actually go and bind to the enzyme on another site, which will change the enzyme shape. Should we know the composition of the macro? Yes, you need to know CHOMPs. So that's carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. Those are the basic elements of all the biomolecules. What are the elements in each one of the macromolecules? Do you think chi-square equation will be on there? I think there may be chi-square, but I'm not 100% sure. I would know it anyway. The elements in each one of the macromolecules. Okay, so let's answer that. You guys, what's in carbohydrates? What are the elements in carbohydrates? Okay, what are the elements in lipids? Good. What are the elements in proteins? Sean, very good. Watch out, Talia. I don't think there's an S in, well, yeah, you're right. There can be an S in some of the proteins. Very good. Because you got disulfide bonds. But they accept Sean as well. What are the elements in nucleic acids? Chomp. Very good. I see some of y'all were like waiting for that. You pressed enter as soon as I said it. Very good. Okay. What other questions do y'all have? I see Kathy, can you upload the PDF forms of study guides? I don't have those. I'm sorry, Kathy. Are you referring to something specific to Fiveable? Are y'all really good? I'm about to go. I have a date with my couch on Netflix. Is 9 to 3 to 3 to 1 ratio only when there is a dihybrid cross? I believe so, but don't hold me to it. I mean, I always say, like, I know you can memorize the ratios, but honestly, I get so paranoid. I just have to work them out every time. When am I going to live stream on DNA? I have actually already live streamed on gene expression. So that's back in the archive if you want to go watch it. So you can go and watch my old videos. They're great. They're exactly like this. It's just me making a fool of myself and talking about science like a nerd because I am a nerd. I'm going to go watch Netflix. And just so you know, I'm just watching reruns of Parks and Rec because that makes me happy. So hope you all have an excellent day. You guys are doing great. Y'all did wonderfully tonight. It was so good to hear from all of you in the chat again, and I'll upload everything later on. Hope y'all have a good day. Bye-bye.